A mouse smells a cat and should flee. An ant loses the trail and should return home. A cricket reaches the edge of dark water and should stop. But now, in the cold logic of evolution, all three do the unthinkable. They move toward danger. So here is the question that opens tonight's audit. When an animal behaves in a way that destroys its own survival but serves a parasite, who is really making the decision? Stay inside that question for a moment. Because it is easy to make this a horror story. It is easy to reach for words like zombie, puppet, possession. And yes, the imagery invites that. A fungus erupts from an ant. A worm drives an insect into water. A virus turns a bite into a delivery system. But if we stop at disgust, we miss the deeper revelation. These are not just grotesque accidents of nature. They are living experiments in agency. They force us to ask whether behavior is as private, as sovereign, as self-authored as it feels from the inside. Picture the mouse first. Night air. A laboratory corridor or a grain-strewn barn, depending on the story you want to tell. The odor of cat hangs in the space like smoke. Under ordinary conditions, that scent is an alarm bell rung by millions of years. The mouse's body should tighten. Its route should bend away. Its tiny heart should vote for survival before thought ever arrives. But infection changes the vote. The expected fear response weakens, or shifts in a way that can make catastrophe more likely. Something small has interfered with one of the oldest decisions a mammal can make. Now the ant. Sunlight fades. Chemical trails thread the ground like invisible roads. Ant society depends on staying in the signal, staying in the system, staying in the colony's collective intelligence. Yet one infected ant breaks formation. It wanders. It climbs. It leaves the ordinary geometry of its life and ascends a blade of grass or a stem with eerie precision. Then it clamps down, as if the world has narrowed to one final command. And the cricket. You can almost hear this one. Dry night, churring in the dark, a terrestrial body built for land. Water is not a destination. Water is a boundary. But the insect drifts toward the shining surface anyway, toward the place where it may drown and where a long-hidden passenger can finally emerge into the environment it needs. Survival for the host ends where the parasite's next chapter begins. What do we call these players in plain language? A parasite is an organism that lives on or in another organism and benefits at that host's expense. A pathogen is a disease-causing agent, often a virus, bacterium, fungus, or parasite, but the important point is the damage. A parasitoid is more lethal still. It uses a host as living real estate and eventually kills it as part of development. And then there is the manipulative parasite, not a formal category in every textbook, but a crucial idea. This is a parasite that does not merely steal nutrients or shelter. It alters the host's behavior in ways that improve the parasite's transmission or reproduction. That phrase can sound abstract, so let's give it an image. Think of behavior as the visible surface of a deep control system. Beneath every action lies a set of ancient switches: approach or avoid, freeze or move, hide or display, sleep or wake, stay on land or seek water, remain with the group or break away. A manipulative parasite may not need to write a new symphony. It may only need to lean on one piano key that evolution has tuned for hundreds of millions of years. This is where a powerful concept enters: the extended phenotype. In ordinary language, that means genes in one organism can create effects beyond that organism's own body. A beaver dam is a classic example. Beaver genes help build the structure in the world. But parasite manipulation is stranger. Here, genes in one organism can produce changes in another organism's body or behavior. The host becomes part of the parasite's expression, as if the parasite's biology reaches outward and writes a line into a different creature's life. Now the mystery deepens. Why do very different parasites, infecting very different hosts, so often produce a surprisingly narrow menu of outcomes? Climbing, biting, freezing, water-seeking, increased boldness, altered timing. Why these patterns? Why not infinite chaos? If each parasite had to understand and re-engineer an entire host brain from scratch, we would expect manipulation to be rare, messy, and wildly idiosyncratic. Yet across fungi, worms, protozoa, and viruses, evolution keeps finding ways to push animals toward a limited set of high-value behaviors. Here is the Audit's first working hypothesis. Manipulative parasites may be converging on a small library of evolutionarily ancient decision nodes. In simpler terms, instead of controlling the whole brain, they may push conserved survival switches sitting near the final output of behavior. Not total domination. Leverage. Not rewriting the self from the ground up. Nudging old circuitry at the moment it matters most. If that is true, we should be able to test it. We can compare manipulated behaviors across many species and ask whether they cluster around the same deep outputs. We can record neural activity in model organisms, stimulate candidate circuits, and see whether activating those circuits recreates something close to the manipulated act. We can compare pharmacology across species and ask whether similar neuromodulators, chemicals that tune brain state like a dimmer switch tunes light, produce similar behavioral shifts. And we can be wrong in a clean way. If manipulation turns out to require unique, whole brain rewiring in each host parasite pair, this hypothesis collapses. If simple circuit activation cannot even approximate the natural behavior, the idea weakens sharply. So what does current science actually know? Let's move from the question into the evidence, case by case, because the details matter. The internet loves one spectacular example and then pretends the mystery is solved. It is not solved, but neither is it empty. Across multiple systems, we have strong reasons to believe that behavior really is being bent toward the parasite's interests. Start with one of the most famous names in this entire field, Toxoplasma gondii. A single-celled parasite with a life cycle that reaches sexual reproduction in felines. Rodents can become intermediate hosts. The broad claim that captured public imagination is simple. Infected rodents may show altered responses to cat odor, potentially increasing the chance of being eaten and returning the parasite to the cat. That story is broadly grounded in real science, but the honest version is more nuanced. Effects vary across host species, parasite strains, tissues, sexes, environmental conditions, and experimental designs. Some studies find striking changes. Others find subtler or context-dependent effects. Still, the central lesson remains. Toxoplasma forms tissue cysts and can alter host neuroimmune chemistry. Neuroimmune means the conversation between the nervous system and the immune system, the brain and body talking in molecular whispers and alarms. If you imagine behavior as a city, neuroimmune signals are not the mayor giving a speech. They are changes in traffic lights, street closures, power distribution. You do not need to command every driver personally if you can alter the system that shapes where movement feels safe or urgent. And that raises a dangerous question. If a parasite can weaken or reshape defensive responses around predator cues, how much of fear is a fixed instinct, and how much is a biochemical setting? Notice what that means. We are not only studying infection, we are studying the architecture of survival itself. Now to the so-called zombie ant fungi, Ophiocordyceps and relatives. This is the image many people know. A fungus invades an ant, compels it to leave the colony, climb vegetation, bite down in a death grip, then kills the ant in a location favorable for fungal growth and spore dispersal. The popular version imagines a fungal pilot sitting inside the ant's brain with tiny hands on tiny levers. But the reality appears more interesting. Evidence suggests the manipulation may rely heavily on distributed chemical control of muscles and physiology, not simply direct occupation of the brain as a puppet master. That matters. Because if the fungus can orchestrate behavior without classic brain takeover, then control in biology is more decentralized than our intuitions suggest. Timing matters. Humidity matters. Height matters. Location matters. The ant dies not just anywhere, but somewhere suited to the fungus's future. We are watching a tiny genome exploit a whole body system whose outputs are already available. Walk, climb, bite, hold. What if the genius of the parasite is not inventing new action, but staging old action under new priorities? That possibility should change how we think about manipulation across the board. Consider hairworms in crickets and grasshoppers. A mature worm needs water to continue its life cycle. The host is a land insect. Under ordinary circumstances, movement toward open water can be rare or actively avoided. Yet infected hosts may head toward water, allowing the worm to emerge. Here too, researchers have pointed toward altered neurotransmission and changed host state. Neurotransmitters are chemical messengers, tiny notes passed between neurons. Change the notes, and you can change what the nervous system finds compelling, frightening, or worth sustaining. The elegance here is brutal. The parasite may not need to turn a cricket into a swimmer. It may only need to convert one fatal threshold crossing into an escape route. A single shift in attraction, persistence, or sensory salience may be enough. Then the lancet liver fluke. This life cycle is so intricate it feels fictional until you sit with the biology. The parasite passes through snails, then ants, then grazing mammals. Infected ants climb grass at dusk, clamp on, and wait where they are more likely to be eaten by a mammal. Dusk is not a decorative detail. It is the kind of timing that makes scientists stop and stare. If the ant remained exposed under harsh daytime heat, that might kill it or disrupt the process. Instead, the behavior is exquisitely scheduled. One parasite lodges in neural tissue while others continue the life cycle after ingestion. So now we face another level of mystery. How does something so small achieve not just the direction of change, but a clock? How does it make behavior happen at dusk, not noon, on grass, not stone, in the cool edge of evening, not in random chaos? The answer likely lives partly in circadian biology, the molecular time-keeping system that organizes daily rhythms. If you can influence the host's internal clock or the pathways linked to it, you can make a behavior not only happen, but happen on schedule. Leucochloridium in snails offers a different style of manipulation. Here the parasite creates pulsing brood sacs that invade the snail's eye stalks, transforming them into bright, moving lures that resemble caterpillars or grubs to bird predators. The snail also tends to move into more exposed areas. This is crucial because it breaks a common misconception. Not all manipulation must be narrowly neural. Change visibility. Change habitat use. Change conspicuousness. Change the odds of encounter. The host does not need to perform a cinematic new ritual if becoming easier to see accomplishes the same transmission goal. And then rabies. Different lineage, different mechanism, same evolutionary logic. Increased salivation, aggression, biting, altered swallowing, behavior becomes transmission technology. If you ever wanted proof that nature repeatedly discovers ways to tie altered behavior to spread, rabies gives it to you with terrifying clarity. At this point, the pattern is impossible to ignore. Manipulation can occur by changing perception, motivation, timing, motor output, or social separation. A parasite does not need to invent a behavior from nothing. It can exaggerate, suppress, retime, redirect, or combine behaviors the host already knows how to perform. Some systems point to direct neural effects, others to endocrine pathways, immune modulation, muscle takeover, or combinations of all three. Endocrine means hormone-based control, the body's slower broadcast system, as opposed to the nervous system's fast wiring. So here is the next unresolved question. Is there a common biochemical language of behavioral manipulation shared across unrelated parasites? In other words, are these organisms all improvising wildly, or are they assembling recurring control modules from a deeper molecular grammar? The Audit's second hypothesis is that parasites deploy what we might call behavioral control packets, modular combinations of neuromodulators, immune-active molecules, and circadian disruptors that can be mixed and matched to bias host behavior. Think of them like compact software bundles. Not a full operating system. Just enough code to tilt the host toward fear reduction, persistence, mistimed activity, social isolation, or water seeking. This can be tested. Use spatial transcriptomics and proteomics, maps of gene activity and proteins across tissues, before, during, and after the manipulated behavior. Use mass spectrometry to identify secreted molecules enriched during windows of control. Feed the resulting data into machine learning and ask whether unrelated parasites with similar behavioral outcomes share recurring molecular motifs. Then try synthetic reconstitution, deliver candidate molecules at naturalistic doses in model organisms, and see whether the target behavior shifts. If no recurring motifs emerge across systems with similar outcomes, the hypothesis weakens. If the observed behaviors are fully explained by sickness, tissue damage, or starvation, without manipulation-specific signals, the hypothesis fails. And that alternative matters. Because science must separate true manipulation from mere pathology. An animal that behaves strangely because it is weak, inflamed, starving, or disoriented is not necessarily being strategically redirected. The strongest cases are the ones where the change is specific, timed, and beneficial to the parasite in a way that exceeds generalized illness. Now we arrive at the edge of understanding, where the pace has to slow down because the paradox becomes sharper. A tiny organism, a tiny genome, an enormous behavioral effect. How is that possible? How can something without a grand blueprint of the host brain still produce acts so specific, so timed, and so effective? Maybe by not doing what we first imagined. Maybe mind control, in many cases, is not command. Maybe it is leverage. Imagine a host brain as a landscape of hills and valleys. The valleys are stable behavioral states, hiding, foraging, mating, fleeing, freezing, exploring. The animal's internal chemistry and the outside world push a ball around this landscape. Most of the time the ball settles into the valley that best fits survival. Now imagine a parasite does not rebuild the terrain. It simply lowers one ridge and raises another. Suddenly the ball rolls into a different valley. The host still performs its own behavior using its own body and circuits. But the probability landscape has been altered. That image helps with specificity. A parasite may only need to move a host from 49% avoidance to 51% approach. It may not need full puppetry if tipping the balance is enough. This is one of the most important conceptual shifts in the entire audit. Once you see behavior as threshold crossing rather than absolute command, tiny causes can produce precise outcomes without requiring impossible omniscience. So here is the third hypothesis. Many manipulative parasites target prediction error and threshold circuits. That phrase needs translating. Prediction error is the brain's way of tracking surprise, the difference between what it expects and what actually happens. Threshold circuits are systems that decide when evidence or motivation is strong enough to trigger action. The idea is that parasites do not micromanage each movement. They alter what the host treats as urgent, safe, attractive, aversive, or worth persisting in. If true, infected animals should not show entirely alien motor programs. They should show shifted gain, shifted salience, shifted persistence. Gain is sensitivity. Salience is importance. Persistence is how long the system keeps pushing a behavior once it starts. Under this model, the host's repertoire stays mostly the same. The weighting changes. Again, we can test it. Use closed-loop neural recordings in infected model organisms to compare sensory valuation and action selection before and during manipulated states. Use calcium imaging or electrophysiology to ask whether candidate circuits show stronger or weaker responses, not wholly new patterns. Perform rescue experiments. Restore normal fear or salience signaling pharmacologically and see whether the manipulation collapses. Build state space models of behavior, mathematical maps of how actions flow over time, and ask whether parasites push hosts into existing behavioral attractors rather than creating brand new ones. If manipulation requires parasite-specific motor programs unrecoverable from normal host repertoire, or if there is no measurable shift in valuation or thresholds before the behavior, then this framework is wrong. And if it is right, the implications spread far beyond parasites. Because then we have learned something unsettling and profound about brains in general. Complex behavior may often be controlled through a few deeply conserved variables rather than detailed top-down command. There is one more scale change we need to make. Most people hear these stories and stop at the individual animal. The ant, the mouse, the cricket. But a manipulated act is not just a private tragedy. It can alter who eats whom, where nutrients flow, how often predators encounter prey, and which life cycles complete successfully. A single behavioral nudge can ripple outward into population dynamics and ecosystem structure. This gives us the ecosystem audit. How much of the living world is being invisibly shaped by parasite-driven changes in behavior? We already know parasites are everywhere. What we do not yet know is how often they act as hidden network engineers, steering food web links by increasing encounter rates among specific hosts and predators. That hypothesis is not poetic speculation. It is measurable. Reduce or enrich parasite prevalence in field systems, and track changes in predation patterns, trophic transfer, and reproductive success. Use environmental DNA and metagenomics to map parasite distributions alongside animal movement data. Build agent-based simulations where parasite manipulation can be turned up or down, and compare the simulated outcomes to real ecosystems. If changing parasite prevalence produces negligible shifts in trophic interactions after controlling for other factors, the idea weakens. If manipulated behaviors do not correlate with realized transmission in the field, the grand ecological effect may be smaller than imagined. But if the effect is large, then forests, fields, lakes, and estuaries are not merely shaped by hunger and competition. They are also shaped by silent contests over behavior. And now the human question returns. What do these systems reveal about agency, about the feeling that our actions are ours? We need precision here, not sensationalism. Parasite manipulation does not prove that free will is fake. It does not erase personhood. It does not mean every urge is an invasion. But it does prove something deeply important: behavior is physically negotiable. What feels like a clean decision can be influenced by molecules, immune signals, hormones, sensory weighting, social context, and ecological pressure. Brains are not isolated computation boxes floating above biology. They are living organs bathed in chemistry, embedded in immune systems, coupled to bodies, and exposed to other organisms that have their own evolutionary interests. The self is real, but it is embodied. And embodiment is porous. There is humility in that realization. And there is wonder. Because once we admit behavior can be nudged with extraordinary precision, we open two futures at once. One is diagnostic. Parasites themselves may become early warning indicators of ecosystem disruption, revealing hidden shifts in food webs before larger collapse becomes obvious. The other is biomedical. The same molecular precision parasites used to bias narrow behaviors might inspire ultra-targeted neuromodulatory therapies, treatments that tune specific circuits more delicately than today's broad-spectrum drugs. That possibility demands ethics as much as ingenuity. We would need molecules with extraordinary selectivity and safety. We would need context-aware models, because what works in a parasite's ecological strategy may be too diffuse or toxic for medicine. But the path is testable. Identify parasite-derived compounds with highly selective receptor effects. Compare them against existing neuroactive drugs for specificity and side effect profiles. Build non-invasive surveillance systems that use parasite presence as a leading indicator of biodiversity stress. If these compounds prove too dirty, too toxic, or too context-dependent, the idea fails. The audit does not worship mystery. It turns mystery into forks in the road and asks which road survives contact with reality. So let's synthesize what we have learned. A parasite can alter behavior not by magically inventing a mind from the outside, but often by exploiting ancient survival switches, shifting thresholds, changing salience, or retiming acts the host already knows how to perform. Across mice, ants, crickets, snails, and mammals, evolution has repeatedly discovered that controlling transmission often means controlling behavior. The deepest clue is convergence. Very different organisms keep finding similar outputs. That suggests the living world contains vulnerable decision nodes, places where a small push can redirect a life. Now carry one thought experiment with you. Imagine that at any moment your own choices arise from countless weighted signals: fear, reward, memory, bodily state, social cues, inflammation, fatigue, hormones, expectation. Which of those feel like you, and which feel like background conditions? If changing a few weights can redirect a mouse near cat odor or an ant at dusk, what does that say about the fragile line we draw between self and system? Then widen the lens one final time. A forest seems still. A stream seems quiet. Yet beneath that calm, spores, cysts, larvae, and viral particles are continuously testing the boundaries of control. Life is not only competing for energy and space. It is competing for behavior itself. If this audit sharpened your sense of what biology is really doing under the surface, subscribe and stay with me, because this channel follows unresolved science all the way to the testable edge. And for the next step, watch the video waiting on your end screen. It opens another frontier where the familiar world becomes much stranger once you inspect the mechanism. Before you go, answer this in the comments. Do you think parasite manipulation is mostly direct command, or mostly threshold tipping leverage? One phrase is enough. I'm genuinely curious where you land.